Okay, so welcome back to this next video in which we are discussing glycolysis. Okay, so we are currently in the payoff phase of glycolysis, and we've reached 2-phosphoglycerate. Okay, so the next reaction that is going to occur is again a reversible reaction, okay, and this is catalyzed by an enzyme known as enolase, okay. And basically what's going to happen is you are going to chop the bond between this carbon and this alcohol group, and again you can imagine sending one electron back to the oxygen and one electron back to the carbon. You're also going to cut this bond between the carbon and the hydrogen here, and again you can imagine sending one electron back to this carbon and one back to the hydrogen. This will lead both of these carbons with free electrons. They will make another bond between the two of them to turn this single bond into a double bond. And then you'll bind this this alcohol group here with the oxygen here with the free electron to this hydrogen with the free electron to create water. So on the forward step, what you'll get is water being given off, basically. Okay, right. And the main product then is going to be what's known as phosphoenolpyruvate. So we've got our double bond here. We've still got these two hydrogens off here, so I'll try and draw it as similar as it was to what I've drawn here as possible. We've then got this oxygen down here, which is linked via a phosphoester link, and I'm going to just rectify this phosphate group. I prefer drawing it this way than that way. Okay, and then we've got our carboxylic acid group here. Okay, and this molecule that we have here now is known as phosphoenolpyruvate. Okay, so that preferentially should be one word, but never mind, I can't fit it all in. So phosphoenolpyruvate. Now, the final reaction then in this entire process is what's known as the reaction catalyzed by pyruvate kinase. And again, this is a reversible, this is not a reversible, this is distinguishable because it's an irreversible reaction. So overall we've now seen 3 reactions which are irreversible. The first one, which was the phosphorylation of the glucose molecule to glucose 6-phosphate by either hexokinase or glucokinase, the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate by phosphofructokinase 1, and finally this one here where we are going to convert phosphoenolpyruvate finally into pyruvate by pyruvate kinase. So let me redraw out the molecule of phosphoenolpyruvate. So here's this double bond. We've got these two hydrogens here. We've got the oxygen with the phosphate group coming off down here. Okay, and then we've got our carboxylic acid group, which will be here. Okay, so this is phosphoenolpyruvate, and often phosphoenolpyruvate is just abbreviated to PEP. Okay, now what's going to happen is an irreversible reaction which is going to involve the removal of this phosphate group here. Okay, and we're going to give it to an ADP molecule again to produce ATP. Now usually when you remove a phosphate group and put it onto ADP, what you would replace it with is an alcohol group. So imagine, as a sort of temporary halfway step, imagine replacing this with an alcohol group. So let's imagine that it's been replaced by an alcohol group. So we've given the phosphate group away onto the ADP molecule to create ATP, and we've then got an alcohol group here. Now, it doesn't stop there. We're going to rearrange this molecule a bit more. So what's going to happen is we're going to break one of these bonds between the two carbons that we just formed. Okay, so we're going to break one of the two bonds. So we're not going to break both. One will remain, but one of the bonds is going to be broken. So send one electron back to each of the carbons. Now this carbon has a free electron and this carbon has a free electron. Then imagine breaking this bond between the oxygen and the hydrogen of this alcohol group that we're imagining is now on there. Okay, and again, this isn't a molecular mechanism, this is just to try and make sense of the reaction. So give one electron back to the oxygen and one back to the hydrogen. Now use this oxygen's free electron to bind with this carbon's free electron to create a double bond between this oxygen and this carbon. So if we keep track of what we've got so far, here is the carboxylic acid group. right on the end here, and that's being unchanged. We've then got this carbon here, which will now have a double bond to an oxygen, okay, and now a single bond to this final carbon, which has got two hydrogens coming off it, but this carbon has a free electron, and we've got this hydrogen with a free electron, so we'll just bind that hydrogen to that carbon. And this molecule that we now end up with is called pyruvate. And again, really what I've drawn is pyruvic acid, but people use them interchangeably. Okay, and the enzyme which catalyzes this final step in the glycolysis pathway is known as pyruvate kinase, and this is irreversible reaction. It can't reverse the reaction. Okay, so that now concludes our discussion of the glycolysis pathway. We've taken glucose and we've turned it into pyruvate. Now, how many pyruvate molecules have we generated? Well, remember we bifurcated the pathway long ago now. Okay, when we split fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, that dihydroxyacetone phosphate was then converted into glyceraldehyde 3-phosphate, and both of these then went through that payoff phase, so both of them will have eventually been turned into pyruvate molecules. So from one glucose molecule we get two pyruvate molecules. Now let's think about how much ATP we actually produce from this. Well, we had to put two ATP molecules in the investment or preparatory stage. We had to put one in the step from glucose to glucose 6-phosphate, and another in the step from fructose 6-phosphate to fructose 1,6-bisphosphate. So we used two, but how many did we get out? Well, in the payoff phase, we got one at this step when we turned from 1,3-bisphosphoglycerate to 3-phosphoglycerate, and another when we go from phosphoenolpyruvate to pyruvate. So we get two, but then we did the payoff stage twice because we had two molecules of glyceraldehyde 3-phosphate. So actually we got four out. So we put in two and got four out. So net production of ATP was two basically. So overall, glycolysis produces two ATP molecules. Now it also produces something else. Remember, we also had the oxidized NAD going to reduced NAD at this step catalyzed by glyceraldehyde 3-phosphate dehydrogenase. And we did this reaction twice, remember, because we had two glyceraldehyde 3-phosphate molecules. So we've also produced two molecules of reduced NAD, which can then go down the electron transport chain route and produce ATP that way. So we've actually produced a little bit more, providing we have the correct oxygen conditions for the electron transport chain to be functioning. Okay, so that now concludes our discussion of glycolysis.